Okay, can you guys hear me?
Yes.
Perfect. All right, anyone have any questions about anything? Okay, so I'm just going to continue on. Okay. So we left off talking about the characteristics of phospholipids in water, and just remember that these are fatty acid tails, and they're hydrophobic because they have nonpolar bonds between carbons and hydrogens. And then we talked about this phosphotidylcholine head, and that phosphate group that we learned in chapter 4. So that phosphate functional group has a negative charge. And remember, water can surround anything with a negative charge. And so that makes this hydrophilic. Hang on a second. Writing tablet's kind of wonky. Okay. Okay, so when scientists learned that there was these phospholipids, oops. When scientists learned that they were phospholipids that made up this cell membrane, and they learned that there are these proteins that were within the cell membrane, they were curious of, and we know about the cytoskeleton, so they were curious of these proteins. Those proteins were anchored in place, or if they were able to move around. So if you can kind of imagine this, the question would be, they have, I don't know if you guys have ever seen on TV, but they have these ship channels and they have these buoys that sort of float in the ocean, and they're anchored to the bottom to sort of direct the large ships to coming into the port. And so the question is sort of, are these proteins anchored? So that means they can't move, they just sit there. Or are those proteins free to just float around in that phospholipid sea, which is that bilayer? And in order to answer that question, what scientists did was they fused. We're gonna skip this freeze fracture, so you guys don't have to worry about this. So when they did this, what they did was they took a mouse cell and they fused it with a human cell. And before they did that, they labeled the mouse cell with some red dye, and they labeled the human cell with some purple dye. I think I'm gonna have to move this tablet because it's kind of weird. And so you can imagine if the mouse cell was, if all the proteins of the mouse cell that are labeled pink here were set in place, and they fused it with a human cell, and all of the proteins here were labeled purple, when they fused the cells together, then you would get just all of the red proteins would stay on one side, and all the purple proteins would stay on the other. But that's not what they saw. What they saw is that the proteins actually mixed together, and you can see that in this image. And that told them that the proteins in the phospholipid bilayer were able to move around. Hang on a second, I'm going to switch my screens here. It's better. So they're able to move around in sort of a phospholipid sea. Hang on. Okay. All right, I got this fixed now. So that means that the proteins aren't anchored, that they can move around in sort of this cytoplasm sea, and so that's kind of interesting. Which they started looking at the actual phospholipids in the cell membrane, and so they can slide back and forth, sort of what it's showing here, and that's what we call lateral movement. They can actually flip from the inside to the outside, and this is mediated by an enzyme. Remember, enzymes are proteins; they're made out of amino acids, and they're like workers in a factory. So they perform a task, and here this enzyme is called flipase. All enzymes end in ASE, and we'll learn this in chapter 8. And so that flipase enzyme can actually flip the phospholipids from the outside to the inside and then back again. This rarely happens, but it still can occur. And so the fluidity, and you guys probably already know this, the fluidity of this membrane, its ability to do these things, flip back and forth and move side to side, really has to do with temperature. It's the same thing with water, right? So imagine if you had a boat or a buoy in the ocean that was able to sort of float around there, but then we froze the ocean. So what would happen to the boat then? Would it be able to move around?
No.
And you also know that that uh, would expand, right? And it would kind of crush that boat. And so it would, the ice itself would change its shape, and that would cause problems for the boat. It would um, change its shape so that it would no longer be able to be buoyant, and then eventually it would sink. And it's the same thing with these proteins that are found in this membrane. So these proteins that go across the membrane, and we call them integral, or sometimes they're called transmembrane. I got a new monitor set up, so it's kind of messing with me. My handwriting looks a lot worse than it actually is. So these are called integral or transmembrane proteins, and usually their job is to transport something either from the outside of the cell to the inside, or from the inside to the outside of the cell. And they're very specific for what they transport. So if their job is to transport water, that's generally the only thing they transport, because the shape is only made to transport water. Or let's say if its job is to transport iron, then it would only transport iron. Or it could transport glucose, and so the only thing that it would transport would be glucose. Now you could imagine if this phospholipid bilayer got too cold, that it would change the shape of this channel, and it would no longer be able to transport things. And that would be bad, right? If you couldn't transport water, that could kill you. If you couldn't transport iron, that could be a factor. Glucose, again, that would be really bad. So you have to maintain a certain consistency of fluid in that phospholipid bilayer. Even though you guys know that you go outside and you're exposed to different temperatures and different elements and stuff like that, if your phospholipid bilayer wasn't able to react to the environment, you would be dead as soon as you stepped outside if it was a different temperature than your body temperature. So thank goodness we have the ability to actually modify the fluidity of our membranes in order to make sure that they stay at consistent fluidity. That, just to give you an idea of what it's like, it's about the consistency of vegetable oil. So if you guys have ever, felt like olive oil or canola oil or something like that, that's about the fluidity of most phospholipid bilayers. And so if you went outside, that would get, and you guys are probably familiar with this, if you took vegetable oil and put it in the freezer, it would get more dense, right? Less fluid. And if you heated it up, it gets more fluid. Same thing with butter, right? So you heat it up, it's more fluid. You cool it down, it's more solid. So we have to maintain this particular fluidity within the membranes. All right, how do we do that? Well, we talked a little bit about this when we were talking about the fatty acids. So we were talking about and we talked about if we put a double bond in there, then that makes it unsaturated. And if we have a single bond in there, it's called saturated. And hopefully you guys remember that unsaturated causes that to bend. So this is the artist's drawing of these bent fatty acid tails. And so they're spread out more. We talked a little bit about snowballs. You wouldn't throw an unpacked snowball. So if it's more fluffy, right, less dense, then it's going to be more fluid. And so this would be an example of the unsaturated fatty acid tails where these molecules are spread out. This is going to be more solid, same word as viscous. And so this would be saturated, whereas this would be unsaturated fatty acid tails. This is more solid. This is more liquid. So unsaturated fats, you guys are probably familiar with this. We talked about it in chapter 5. So usually these are plant-based, and saturated is usually animal-based. So plants are able to add unsaturated fatty acid tails if they get cold. So like a good example of this is winter wheat. So winter wheat changes its fatty acid tails depending on what the temperature is outside. So you could imagine if it gets colder, so if it's cold outside, then what would you need to do to maintain the fluidity of your membrane? Would you need to increase the fluidity or decrease the fluidity? In other words, if it's cold out, would you need to make it more liquid or more solid?
More liquid.
Yeah. And to do that, you would have to do what? Add saturated or unsaturated?
Unsaturated.
Exactly. So if it's cold outside, what winter wheat does is it replaces the saturated fatty acid tails with unsaturated ones to make it more fluid. And when it gets warm outside, so in the summer, would you want to make it more liquid or more solid? So you're heating up.
Solid.
Yeah, you want to tend to make it more solid. And to do that, what you would do is just replace these uh, unsaturated fatty acid tails with saturated ones. Um, And that way you can maintain that fluidity regardless of what the temperature is outside. And this is a trick that plants do because they're not able to change their temperature. They're not warm-blooded like we are. Now, animals, we don't really have a lot of unsaturated fats. And so most of our fats are saturated. And again, we talked about this in chapter 5. And so we can't really do this. We can't really change the fatty acid tails between saturated and unsaturated. This is for plants, but animals, instead of doing that, what they do is they use cholesterol. So this molecule, in case you don't recognize it, we looked at it in chapter 5, and it's that 4-ring molecule that is cholesterol. And so that's what the artist is sort of drawing in here. So these yellow things are cholesterol. And what cholesterol does is it actually goes into the phospholipid bilayer, right? Because it's a fat. And so fat, like dissolves like. And so it can get into these fatty acid tails. And it can actually physically bend them. So you can see that it's physically causing the tails to bend. And when it does that, it spreads them out. And if so, you can change the fluidity of your membrane, even if you're an animal, by changing your cholesterol, the amount of cholesterol in your cell. So let's do this little game again that we were just doing. So this one was for plants. Now let's talk about animals. Okay, so let's say that you guys go to Minnesota in the middle of winter. So it's very cold out. What would you want to do? Would you want to make your membrane more fluid or less fluid?
More fluid.
Yeah, because if it's cold out, it's going to get more solid. And so you need to make it more fluid so you keep that consistency of vegetable oil. To do that, would you need to add cholesterol or remove it?
Add cholesterol.
Yeah, because it's going to bend those membranes and spread them out and make them more like unsaturated. And so if it's cold outside, animals add cholesterol to their membrane. And if it's warm out, let's say in the middle of Phoenix in the summer, then you want to make your membrane more fluid or less fluid?
Less fluid.
Awesome. And so we would want to remove cholesterol from our membranes. So cholesterol, even though it's got a bad rap, is really important, right? We talked about it. You need it to synthesize pretty much all of your hormones, and you also need it to maintain the fluidity of your membranes. If you didn't do that, then the proteins in your membranes would change the inside of those channels and wouldn't be able to transport things that you need, like sugars and stuff like that. All right. So we kind of covered this. Membranes have to be around the consistency of vegetable oils. So that just gives you an idea. Cells, they can change their composition of their phospholipids to deal with changes in temperature. And then we talked about plants do it by changing their unsaturated phospholipids, and animals do it by adding or removing cholesterol from their membranes. Okay, so let's go back to looking at a membrane. This is from your book, and it's just an artist rendition of what a membrane looks like. So that, we normally draw it like this, but it's really complicated, right? We have the bilayers. I'm not going to draw 2 layers in there, but we have proteins that go across, and they transport things like glucose. They transport things like water, and remember, they're all different. So that's what these things are showing. And because they go across the membrane, they're called integral. So they're integrated into the membrane. And if they don't go across the membrane, if they're on the either side of the membrane, then they're called peripheral. Like if you think about your peripheral vision, where does that? In the front or on the side?
The sides.
Right. So just think about that. The proteins that are on the inside or the outside are called peripheral, and the ones that go through are integrated into the membrane, and they're called integral. So you just need to know these terms, make you some flashcards or whatever, and you just got to memorize it. So integral proteins go across the membrane. They're trans. Remember, trans is across. They have hydrophobic regions that span the hydrophobic interior of the membrane. So remember, these are proteins, and remember, proteins have those R groups, and the R groups here are going to be inside the membrane are going to be polar or non-polar. Remember, like dissolves like. So what's the inside of the membrane?
Is it polar?
Uh,
Non-polar?
yes, non-polar. Because the inside of the membrane is non-polar. It's hydrophobic. So that's how it's able to go through here. And then, what the artist isn't showing you is there's channels in here that allow things to move through it. Those channels have a specific shape that dictates their function. And if those channels change their shape, they would no longer be able to transport these things from the outside to the inside. You have other things that are transported out. And we're going to talk about, oxygen and stuff that can go across the membrane without a channel, and why that is. And then we have peripheral proteins, and we just talked about those are attached to the surface. They don't go across. Like this is a peripheral protein. All right. The membrane is asymmetric, which means A means not. So you can see that the outside of the membrane, remember this is the extracellular matrix, is very different than the inside of the membrane, which is the cytoplasm. All right. So integral membrane proteins we talked about, they have a variety of functions. We talked about transport. So, glucose, water, whatever, it goes through these channels. Sometimes it requires energy to transport it. Sometimes it doesn't. We can have, so these proteins can have specific areas where they do reactions. So they can have enzymes on it. We talked a little bit about like maybe you want to do a hydrolysis reaction. So you would have a protein area here that is going to be attracted to water. Remember hydrolysis, you have to add water to break molecules apart. And so you would want an R group here that would be hydrophilic to attract water to it. I'm still getting used to this pen, so forgive my handwriting. Some things don't have to go across the membrane. So insulin, human insulin is like this. Human insulin never actually goes into the cell. It just binds to the outside, to the integral membrane protein, and that changes its shape. When we put one molecule to another molecule, it changes its shape. And remember, once we have a shape change, we can get it to make a signal. It's kind of like if someone knocks on your door or rings the doorbell, they're there without even having to open the door. These integral proteins can join cells together. So if you look at a blood vessel, you have cells that are bound together like this. And so that keeps your blood inside. So this is a blood vessel. And these are individual cells that they're smooth muscle cells. You'll learn this. You'll learn more about this when you take anatomy and physiology. But your blood vessels are lined with smooth muscle cells, and the smooth muscle cells' job is to keep your blood in your bloodstream, right? You don't want your blood leaking out. But there are things in your blood that need to go across this cell, this intracellular junction. And so let's say you get an infection, well, your white blood cells are inside your blood. And so these need to open up a little bit. So the cells spread out a little to allow these white blood cells to go through the blood vessel and then to go to your other cells to fight infection. So these have to respond to the environment, and like the allergies, which is histamine. So histamine is released when you have allergies. Um, histamine's job is to relax, so we call that a vasodilator. And so this is why people go into shock and their blood pressure drops, because you have this vasodilation where these intracellular joining regions are relaxed and too much blood comes out of your blood vessel. It would kind of be like if you poked a bunch of holes in your garden hose, you wouldn't get enough the same amount of pressure out the end. And so that can cause, anaphylactic shock and things like that if you have too much histamine. There are also things on the cell surface that allow cells to recognize other cells. You probably know that your heart cells hang out with other heart cells and your brain cells hang out with other brain cells. So it's not general, unless you have cancer or something, but this is really important in cancer biology. So generally your lung cells would stay in your lungs. They wouldn't just go, okay, hey, I'm going to go visit the brain today. If they did that, then it would mess up your brain cells because lung cells have a different function. So they would get in between your brain cells, and it would be similar to, shoving an ice pick into your head or something like that. That's what happens with cancer. They lose this cell recognition. And so lung cells don't realize that they're supposed to be with other lung cells, and they travel off to a different area. You can kind of think of it as area code. So let's say if I change 602 to 623, and we'll say that these proteins here are an area code. So this is 602, and I change this, well, now it becomes 623. And so the cell doesn't think that it's supposed to be in Phoenix proper anymore. It thinks it's supposed to be on the west side instead. And we've only changed one number, right? So that's what happens in cancer when it spreads. This cell recognition fails. And this is really interesting for cancer biologists. And then the other role of these integral membrane proteins, although it's more rare, is to attach to the cytoskeleton and the extracellular matrix. This is the most common. And this is what we're going to focus on in this chapter. Any questions so far? Okay, so we talked about homeostasis, and you guys know that homeostasis is super important. In order to maintain homeostasis in this membrane, the cell has to select what comes across and what doesn't. So we call that selectively permeable. It's the property of membranes that allow some things to cross more easily than others. This really depends on the solubility of membranes that we talked about earlier, how fluid this is. And it also depends on the presence of these transport proteins, these integral membrane proteins like shown here, that, you know, in this case, it's transporting this green box, which maybe it represents glucose or something like that. We'll just say it's glucose. Okay, so, but not all things have to go through a channel. Some molecules, like let's say that this is oxygen. So oxygen, if we look at oxygen, you guys know oxygen is, this is from way earlier, chapter 2. Oxygen doesn't like to be alone because it only has 6 electrons in its outer shell. And so it has to form 2 bonds. So anytime oxygen is found with other oxygens, it forms a double bond. And so from your experience, you should be able to tell me, is this polar or non-polar? In other words, is oxygen sharing equally or unequally? Remember that all depends on electronegativity. So if you have 2 oxygens with the exact same strength, are they going to share or not?
Yes.
Yeah, and they're going to share equally because they have the exact same strength, right? 3.5 minus 3.5 is 0. So if they share equally, are they polar or non-polar?
Non-polar.
Yes, exactly. And what's this membrane in here? Remember, these are carbons and hydrogens. They're also non-polar. Remember, like dissolves like. So here's the question. Can oxygen dissolve in the phospholipid bilayer?
Yes.
That means that oxygen doesn't need a channel. It can go freely across this membrane because if it was water, it would be scared away, right? Water and fats don't mix, but oxygen and fats do. So oxygen can go through this membrane without a channel, whereas water couldn't. So here's the rule of things that can go through the channel. I'm sorry, through the membrane with a channel like this, or without a channel like this. So small molecules that are hydrophobic, right? Hydrophobic means they're non-polar, or electrically neutral. Remember, water cannot dissolve anything that has no charge. So anything that's electrically neutral is going to be hydrophobic, right? Because water, in order to be hydrophilic, you have to have some sort of charge. So it has to be hydrophobic, electrically neutral. If that's the case, small, hydrophobic, electrically neutral, it can pass through the plasma membrane by dissolving across it, right? So oxygen, the example I used was oxygen. Oxygen can pass through that membrane no problem without having to go through a channel. Whereas water, does water fit into this category?
No.
Yeah. So water's small. I'd say it's a small molecule. It's only 3 atoms. But water is not hydrophobic. Water likes itself, so it's hydrophilic. Um, it is also not electrically neutral. Remember, it has those sigma charges on it, those sigma positive and sigma negative. So water cannot pass through the membrane without a channel. Glucose, we can look at glucose. So glucose, I would say it's not a very small molecule. Um, it does sugar dissolve in water?
Yeah.
Yeah, so then it's hydrophilic. It's not hydrophobic. So glucose would have to go through a channel. Water would have to go through a channel. It doesn't fit those rules. So for the test, make sure you guys know the rules. Small, uncharged, hydrophobic. And these are pretty much the same definitions. But anyway, those are the three rules that allow things to pass through the membrane without a channel. And you should know those. Okay. So because these things are not hydrophobic, things could go through without a channel, right? So because they have to go through this channel, usually those molecules that go through the channel are going to be hydrophilic. So the channel itself has to be hydrophilic because it has to dissolve hydrophilic molecules. It can physically move substances across a membrane, and I'll show you how that works as we go on. And each protein is specific for the thing that they transport. So remember there's a integral membrane protein called an aquaporin, and it wouldn't be called aquaporin if it transported anything except water, because aqua means water. So aquaporins don't transfer sugars, they don't transport iron, they don't transport anything, sucrose or fructose or anything else, no proteins. They only transport water. So everything is very specific for what it transports, and that's really important. Okay, so that brings us to what you guys did in your lab, your last lab, which is osmosis. So osmosis really relies on this process called diffusion. You guys are probably familiar with diffusion. Diffusion means that molecules go from an area of high concentration to low concentration. So if you had something super stinky in your trash can, the closer you got to the trash can, the more stinky it would be. And the further away, the less stinky, right? So high to low. If you dropped, so let's say you had a glass of water and you put a drop of food coloring in it. So let's say we just say blue food coloring. Or maybe I should use orange because that's what the artist is using here. So we put a drop of orange food coloring in here, or blue, it doesn't matter. And you put this on the counter and then you came back like 6 hours later. Would that drop of blue food coloring still be hanging out in the same place?
No.
No. And it's because, on a molecular level, the water molecules are moving around, right? They move. And so they actually act like a mixer. And they're going to mix this stuff around so that everything is even. Now, the artist has drawn these molecules of dye. Let's say that there's a membrane. That membrane allows the dye molecules to pass across it. So we have a high concentration of dye on this one side of this membrane. And over time, the dye molecules are going to move because they fit through there to the other side until everything is mixed evenly. So when you came back, it wouldn't be like, part of your cup was blue in the corner or orange in the corner, right? It would be all mixed evenly. And so if the artist drew this right, there would be an equal number of molecules on each side. And then we can count them 1, 2, 3, 4, 5, 6, 7, 8. There's 8 on this side. There should be 8 on this side. 1, 2, 3, 4, 5, 6, 7, 8. There's 8 on this side. And so everything is mixed evenly across that membrane. And that's what diffusion is. A high concentration to this side's low concentration. There's no dye molecules here. Low, high. So things move from high to low. And we call that a concentration gradient. That means it goes from high to low. It's just a term scientists use. And remember, this results from the kinetic energy of the molecules. The water molecules move by the definition of temperature is kinetic energy, right? And the more kinetic energy, the higher the temperature. The less kinetic energy, the lower the temperature. And you can go all the way to absolute zero. which is really cold, minus 400 degrees plus. And that at that point, then molecules will stop moving. But anything above absolute zero, and even the darkest reaches of space still have a temperature. So you're no matter what, you're always going to have molecules moving at some rate or another. And it may take, like, if you put a drop of food coloring in ice, it may take 1,000 years for it to distribute evenly, but it will eventually distribute evenly. All right. So do you guys have any questions about that? So the last thing I want to say is that it's not affected by anything else. So if I put a drop of blue food coloring in this water and also yellow food coloring in this water, the diffusion of yellow does not depend on the diffusion of blue. In the end, they're all going to diffuse equally, and we're going to end up with a green cup of water because blue and yellow make green, in case you didn't know that. All right. So we call this passive because the energy is already in the system. The kinetic energy of temperature is already in the system, right? As long as there's temperature, there's going to be kinetic energy because we define this in chapter 3. We talked about the concentration gradient, high to low. We're comparing two areas in the solution, high, low. And like we said, it's passive. The energy is already there in the kinetic energy of just because there's a temperature above absolute zero. And there's going to be a net movement. So here we have the artist is drawing like this red and this orange. So these things, as long as these holes are big enough to pass the dyes through, then they're going to move through there and they're going to move evenly. This is high, so this is going to move this direction. And this is higher than this side, so these orange ones are going to go that direction. And in the end, if we wait long enough, we should have equal numbers. So let's see, 1, 2, 3, 4, 5, 6. There's 6 purple ones and 3 orange ones on this side. And we do the same thing, 1, 2, 3, 4, 5, 6. There's 6 purple and 1, 2, 3 orange ones. So they're mixed evenly. And this just takes time, and it changes due to the kinetic energy. So if the temperature is higher, this diffusion will occur faster. If the temperature is lower, the diffusion occurs slower. That's why you sort of heat up stuff when you want to, say dissolve sugars or whatever into water. You can increase this diffusion, the mixing of things, by changing the temperature. Any questions so far? Okay, so let's go back to the cell. We have this cell. We know that water can't pass across this membrane, but your cells have these things called aquaporins, which are water holes. Aqua is water, porn is hole, and they're always present. So that means when you have water outside of the cell, it's free to go in or out of the cell through these aquaporins. It can't go across the membrane. So I might ask you on the test, can water pass across the membrane without a channel? And the answer is no. And I might ask you, what's the name of that channel? You should be able to tell me it's an aquaporin. So osmosis is a phenomenon that results from diffusion and the fact that we have these open channels in our cells all the time. So the definition of osmosis is diffusion of water. Remember, it's just water across a selectively permeable membrane. In this case, it's the plasma membrane. And that's due to the differences in the concentration of solutions on each side of the membrane. So that's kind of a wordy definition, but let's just think about it this way. So let's say that you are shipwrecked and you're in the ocean, and so this will be my person. Probably not smiling because they're shipwrecked, but whatever. And they have a stomach, right? And that stomach is here. And that stomach is lined with cells. So I'm not going to draw the whole thing, but your stomach is lined with cells. And let's say you're going to drink the salt water. You probably don't want, no, you don't want to do that. And salt water is about 4% salt. So you've done this. You've drank the salt water. And so let's say this is one of these cells. We're just going to look at one of these cells. And so that means on the outside is 4% salt. The average salinity of your cells is 0.9% salt. So this is the inside. This is the outside. So which one is more salty, inside or outside?
Outside.
Right. And so remember, these aquaporins can't transport salt. They can only transport what?
Water.
Yeah. So what's going to happen is the salt can't move. It's stuck because it doesn't have a channel. The membrane would have to build the channel to transport salt. And it doesn't want to do that because it needs to maintain this amount of salt inside the cell. But its fault is that it has these water channels and water can move. So based on diffusion, water is going to want to try to make these equal, just like we showed before in this slide, right? Everything wants to be equal. And it doesn't matter. Let's say that this is water and this is salt. It doesn't matter. They're all going to try to mix to be even so that there's same amount, the same percentage of salt on each side of that membrane. Okay. So if that's true, what direction would water need to go to try to make this 0.9 equal to this 4?
It would go out.
Yeah. So imagine if I had a glass of really salty water and I wanted to make it less salty, right? I want this to go down. I need this to go down to get it closer to this 0.9. Then I would pour water in it, right? That makes sense. Like in not on a cell level, but a normal level. If I had really salty glass of water and I wanted to make it less salty, I would add pure water. That makes sense. Now, if I wanted to make my salty water more salty, because these want to get into to be a happy medium. Let's say, I'm not going to do the math, but let's just say the happy medium is 2%. This wants to get to 2%, and this wants to get to 2%. So how do I make this more salty?
Take away the water.
Yeah, I remove water. So this take the water coming outside of the cell from inside is just following the rules of diffusion to try to make this more salty on the inside of the cell and this less salty on the outside of the cell so that they can become even. And because this concentration of, because the cell is almost always the same inside, we usually refer to the concentration of salt on the outside of the cell. So we use the term hyper, iso. Remember that word iso is same. We learned that from isomer and we learned that from isotope. And so isotonic, this is the tenacity of the, or the saltiness of the liquid. Isotonic would mean it's the same, right? Iso, that word iso is same. So we have hyper. So you guys know what that means? Like if you're hyperactive, you're highly active. If you're iso, that's the same. And then you could be hypo. And what would hypo mean? Like if you're hypoglycemic.
Low.
Yeah. So these words are used in osmosis to describe the concentration, unless you're told otherwise. So sometimes you're told otherwise, but unless you're told otherwise, this refers to the concentration on the outside of the cell, right? So go back to us drinking salt water. Why don't you want to drink seawater? What's going to happen to the water in these cells and in your body?
They're gonna go out of the cell.
Go out, and it's going to increase the amount of water that comes out your butt, and so it's going to give you diarrhea, and it's going to make you dehydrated because it's sucking the water out of you because of osmosis, right? And so this example that we just used, the salt water is higher on the outside than the inside. So that would be hyper, iso, or hypo.
Hyper.
Sweet. Hypertonic. That's awesome. That's exactly right. Now, there's other situations, and these are real life ones, and I kind of talked about it in the lab. So there, when the Nintendo Wii came out, this happens a lot in like fraternity and sorority hazing. Sometimes they'll make people just drink a ton of water, and a lot of times people die from that. So you would think, oh well, you could drink as much water as you want and you're fine, and that's right. So, but it also can be wrong, and I'm going to explain to you why that is. So here's my stick figure, right? And this stick figure is going to drink a lot of water, right? And you have kidneys, and your kidneys' job is to regulate the amount of water in your blood, right? So um, if you drink a lot of water and you pee it out, then this balance is going to it's going to be maintained. But what happens in these deaths is that people drink water, but they're not allowed to go to the bathroom. So that contest for the Nintendo Wii was hold your pee for a Wii. Um, and so people would drink water, but they weren't allowed to go to the bathroom. And this sets up a unique situation where your kidneys can't do their job. And what's happening is that you're increasing the amount of water in your cells, well, in your blood. So here's our cell. Remember, it's 0.9% normally, and your blood, which is going to be on the outside of your red blood cells, right? It's your blood's made out of liquid mostly, and proteins and stuff like that. But if you keep drinking water, your blood would normally be 0.9%, but I'm pouring water into 0.9%. What's going to happen to this number? Is it going to get higher or lower?
Lower.
Yeah. And so let's say it's close to 0.1% because you're drinking this water and you're not peeing it out. Your blood is going to have a lower concentration than, say, your red blood cells that are floating around in your blood. And so what's going to happen to the water here? Remember, it wants to be equal. So if to be equal, does this have to, the 0.9 have to go up or down?
Down.
Down. And to be equal, does 0.1 have to go up or down?
Up.
Yeah. And so what does that mean? What's the direction of the water movement then?
It's gonna go into the cell.
Exactly. So the water will go into the cell, and that's super dangerous because now you're adding water into a cell. It's the same thing that if you were adding air into a balloon. If you keep blowing up the balloon and blowing up the balloon, what happens to the balloon eventually?
It's gonna pop.
Yeah, it'll burst. And if your red blood cells burst, they can't carry oxygen and you will die. So hold your breath, right? We talked about this. And your brain cells will also burst, and all your other cells will burst. So you don't want to have a weird concentration of salt inside your blood that's different than what's inside your cells. And so I don't know if you guys have ever been to the hospital or whatever, but I'm sure a lot of times that you go there and they're like, oh yeah, you're dehydrated or whatever. We're going to give you an IV, right? So do you think that IV would be pure water?
No.
Yeah, because if they gave you an IV of pure water, your cells, your red blood cells would swell and burst because pure water would be zero. Oh, I erased that. Pure water would be 0% salt, and you'd still have the same effect. Water would rush into your red blood cells and cause them to swell and burst. So what do you think the percent of salt they would use in an IV?
Would be 0.9.
Yeah, you want it to be the same so that water doesn't come out and dehydrate your cells or go in and cause them to burst. So IVs are 0.9% salt to match what's in your cell. And they never vary. So, if you went to the hospital, or maybe you guys work in the hospital or in the medical profession, look at the IV bag. It always says 0.9% sodium chloride because that is what we call isotonic to your cells. The same, 0.9 on the inside, 0.9 on the outside. So make sure you guys know these terms: hyper, hypo, and iso. And remember, so this condition right here, where the salt concentration is lower on the outside than the inside, is hypotonic, lower outside. This is higher outside. And that's by default. So in some instances, I might say, what's the solution relative to the inside of the cell? And so in this case, the outside of the cell, it would be hypotonic, right? But if I'm talking about the inside of the cell, the inside is higher than the outside, so that would be hypertonic. And so when you guys take your test or your quizzes, make sure that you pay attention to what the question is specifically talking about, the inside of the cell or the outside of the cell. If it doesn't say, it's always the outside of the cell. All right, so this is just showing you that it doesn't matter. So in this case, this dye cannot move, these molecules of dye cannot move across this membrane. So they're stuck. So what has to move? Water. And so you can actually get water to defy gravity through osmosis, but this osmosis is actually stronger than the force of gravity. So you can get these U-shaped tubes to increase the height on one side and decrease the height on the other. All right. This is just showing you that across a selectively permeable membrane that is porous to water, water is going to move out and in at an equal ratio, which we already talked about. So these are some terms that you guys need to know. Osmotic concentration is the concentration of the solute. Remember, we learned solute is the material being dissolved. The solvent is the material that's dissolving. And then the solution is the two together. So this is the concentration of the solute. If we're talking about salt water, the osmotic concentration would be the concentration of salt. Like we talked about, 4% for seawater, that would be the osmotic concentration, or 0.9% for your blood. That would be the osmotic concentration. The osmotic pressure is the pressure exerted by the water flowing in or out into, and this means semi-permeable membrane. It's just an abbreviation that scientists use. So if we were in a hypotonic solution, like we talked about with drinking too much water or putting water directly in your blood vessels, that would cause this to go in, and the pressure would increase, just like the pressure in a balloon increases the more air you put in it, right? The balloon's harder to push on the walls. And so this pressure that's created by water movement is called osmotic pressure. And then reverse osmosis works, it's pretty simple. So you have a membrane that's only permeable to water, and you have your dirty water on the outside, and you have a high concentration of salt on the inside. So if you have a high concentration of salt on the inside and a low on the outside, which way is the water going to move?
Out. Or in.
Yeah, it wants to make this less and this more. So the water is going to go in, and all of the dirt and stuff won't be able to go across that membrane, only water. So what you're doing is you're increasing the water in here, increasing the pressure, and then there's like a piston that can push against this. And since it only water can flow through that, in the end, pure water comes out the top of this membrane, and that's what you drink. So in Arizona, because the water is so gross, it has so much dissolved solids in it, that it's very common that people will drink reverse osmosis water here. And so it's not magic. It's just putting high salt across a semi-permeable membrane, just like your cell, and then overcoming that osmotic pressure to force the water to come out of the membrane to be pure water. And that's how those machines in front of the grocery store work, that you put the quarter in and get your gallon of RO water. All right. So we talked about animal cells. We know that animal cells like to be in an isotonic solution. So the concentration on the outside is the same on the inside, because we don't want the cells to shrink or burst. If we put too much water in our blood, what happens to our blood cells? They get full of water and they burst. So hypotonic solutions, the water goes in. And then if we drink salt water, like seawater, the water is going to come out of our cells and cause them to shrink or be dehydrated. So the lesson of this is that animal cells want to be what? Hyper, hypo, or iso?
ISO.
Yeah, exactly. But plant cells are different. So plant cells, because they have this cell wall around them, having some pressure on the inside actually causes each of the cell walls to push against each other, and that causes more strength of the whole thing. So plant cells like to be a little extra full, and so they like to be in a hypotonic solution. If you don't water your plants, what do?
They die.
Well, what do they do first?
Or they dry out.
Well, before that.
They get soft.
Yeah, they will, right? So they will get soft. They start bending. So that's because they're not hypertonic. Remember that we have water on the outside. You water your plants. The concentration of water is higher on the outside than on the inside. So water is going to move in, and that's going to cause them to be rigid and stiff. So when you're not watering your plants, they start to wilt. Eventually they'll dry out and they will die. And if you put plants in a hypertonic solution, the plants have a cell membrane that's attached to the cell wall at different points. And so this is made out of cellulose and it's really hard. And what happens is that the plasma membrane will shrink, and these attachment points, it could tear. And if it tears, it would tear the membrane and it wouldn't be able to maintain homeostasis. So if you put salt water on a plant, it could destroy that membrane and cause it to not be able to maintain homeostasis, and then it would permanently die. Whereas if you have wilty plants, you could just add water to them and they would come back. We call that plasmolysis. Remember, lyse is to break apart, and plasmo is referring to the plasma membrane. So that occurs in hypertonic solutions of plants. So in animals, let's just summarize. In animals, what do they want? Hyper, hypo, or iso?
ISO.
And in plants, what do they want? Hyper, hypo, or iso?
Hypo.
Perfect. All right. So we've covered osmosis, and now we're going to talk about how things go across the membrane. So we have something called facilitated diffusion. Remember, not all things can go through this phospholipid bilayer. The rules are it has to be small, and it has to be uncharged, and it has to be hydrophobic. Otherwise, it needs a channel, like we showed right here. So this molecule does not fit this description, and therefore it needs a channel. Now, this channel could be open all the time, like the aquaporin we talked about. So let's say that this is water. Water can't move across the phospholipid bilayer. It needs that channel. And so it's going to move from a concentration to high to low through diffusion, right? We just talked about that. So that means there has to be more water molecules on the outside of the cell than the inside. This is the inside. This is the outside of the cell. And if that's true, then water will flow in. If there's more water on the inside than the outside, then water would flow out. This is passive. Remember, the energy is in the energy is in the kinetic energy, the temperature. And that this channel helps these molecules that couldn't cross the membrane to be able to cross the membrane. So we can have a special kind of facilitated diffusion where the channel isn't open all the time. It's actually gated. And one thing that you guys should remember, one of the most important things to learn in biology is that if I take a molecule, whatever this might be, like we could say that this is glucose. If I take glucose, and this is a protein, remember, so this is made out of amino acids. And if I add a molecule, attach it to another molecule, what does it do to that molecule? And the answer is it changes its shape. That's really important. Anytime I add a molecule to another molecule, it changes its shape. So if this glucose binds to this channel protein, it's going to change its shape. And in this case, it's open on the outside, but when glucose binds to it, it's going to be closed on the outside. That changes its shape, and that causes it to be released on the inside. So it's still concentration of high to low. High on the outside, low on the inside. So there's no energy. It's still passive. But this is gated because it sort of regulates what rate things come in and out. So like we said, still passive, no energy required. All right, so passive transport is kind of like if you're on a mountain and you poured water on the top of that mountain, where would that water go? Up or down?
Down.
Right. And that's because of gravity, right? Gravity gives us free energy. That's how dams work. Water moves from high areas, elevation, to low elevation, turns those turbines and gives us energy thanks to gravity. And it's free, right? Because gravity always exists. Um, this is kind of like passive transport. Temperature always exists. So we're going to get free energy out of the temperature, and we can use that to move stuff across the cell. But there's times where we want to move the water up the hill. And so how do we move water uphill?
Energy.
We would need some sort of energy. So like if you wanted to carry a bucket of water uphill, you would need energy to do that. You would have to physically climb that hill, and it might be tiring, and you could tell that you're using more energy than normal by walking that water up the hill, as opposed to pouring it downhill, which wouldn't cause you to use any energy at all, right? Um, so, or you could pump it, right? You could use like an electric pump or whatever. But in whatever case, if you want to go against gravity, you have to put in energy, right? If you want to go against the concentration gradient, you have to add energy. And so when we add energy, we call that active transport. It's the use of energy to transport a molecule against the concentration gradient. So that energy, we know our energy source is usually ATP. Sometimes it's called pumping, just like pumping water up a hill. And it helps maintain gradients that are used to do work in the cell. So I'll just finish this up and then I'll jump over to office hours. But here's an example of active transport. So in your body, you have a thing called a sodium potassium pump. We'll start here. This is how the artist did it. So this is just showing you time, like different snapshots. So this would be first and second and third. It's not really the cell, but it's just showing you. So uh, we have, we'll start here. So in the cytoplasm, you have sodium. Sodium can bind to this sodium potassium pump, which is, remember, this is a protein in purple. And sodium, you guys know, is an element like that makes up table salt. And so it'll bind 3 sodiums. And you can see the artist has drawn the shape of the protein as this little, half circle. So circles fit into half circles nicely. Now, when we take ATP and we add a phosphate, so it becomes ADP, which is adenosine diphosphate. This is triphosphate. So it's giving up a phosphate, adding it to this molecule. When we add a molecule to another molecule, what does it do to it?
Changes its shape.
Yes. And so you can see right here that when we add this phosphate, these are no longer nice circles like they were here. They're kind of pinched. And this is no longer a super pointy triangle. It's more of a real, a less steep triangle. And so the sodiums are forced out by this phosphate's presence. And so they go out. So we're moving it out of the cell, sodiums. And now potassium, which the artist has drawn, is a different shape, which it really is. Maybe not a triangle, but it's a shape. Now these potassiums can bind to couldn't bind to the steep triangle, but it can bind to this not so steep triangle. The potassiums bind. We remove the phosphate. So what does it do? It goes back to its original shape, which pinches the potassiums that go in. So we're moving sodiums out 3, and each charge on a sodium is what? +1. So we're moving 3 positive charges out of the cell. Potassium is moving in, and we're moving 2 potassiums. So what's the charge on potassium is +1. So we're adding 2 to the inside of the cell and 3 to the outside of the cell. So what do you think happens to the outside of the cell? It gets positive charges, and on the inside of the cell, it's negative charges. And this works just like a battery to generate electricity, which you use to do work inside your body, right? Your body uh, invented the movement of electrons and electricity long before Benjamin Franklin flew his kite. So this is basically how you live, by generating this electricity. And this moves your muscles and can do other cool stuff. So that is a form of active transport. By using ATP, we're pumping more sodiums out than potassiums in to generate this electrical current. We call this an electrical potential because a battery would have potential energy, right? Um, until you plugged it into something. So that's why we call it electrical potential. And then you release that to spin motors or move your muscles or whatever. All right, any questions? All right, so I'm gonna wrap it up here. You guys have a good weekend. I will have office hours next. And if you don't show up there, then I'll have office hours again on Monday, back to the regular schedule. And then remember, next week is the last week before spring break. So you guys will have a week off, and so will I. And then we'll pick it up when we come back after spring break.
Thank you.
All right. See you later.